In this introductory lesson, DNA to protein, what I want to do is lay out the course, basically. Go through each of the different sections that takes us from DNA and gets us to amino acids turning into proteins, proteins doing things with kinetics. I also want to talk about how prokaryotes differ than eukaryotes. We're not going to talk about RNA organisms, viruses, retrovirals. We're not doing that here. We're going to be focusing on double-stranded DNA. And prokaryotes are going to serve as sort of the foundation since they're simpler. And then we're going to spend a lot of time talking about the complexities of the eukaryote. So everything is going to be based upon DNA, the genetic code for which everything our body does is based. And what I hope to instill by the end of the lesson is just how powerful DNA is. Our cells all have exactly the same amount of DNA, the exact same genetic code in all of my cells, no matter whether it's an hepatocyte, a neuron, a myocyte, whatever, the DNA is all there. Which means that we had to devise some method of storing all of that DNA in one place without it getting in the way of the function of the cell, which means we had to package and store it really well. We're going to begin the course by talking about DNA. We'll talk about nucleotides, nucleic acids, forming bonds between them, the structure of DNA, the helix, and the like. Then we'll move into the complicated process of taking whatever DNA you've got and making an entire another set. That is the process of replication. Replication is difficult because all of the DNA must be copied at the same time while also protecting the DNA from degradation. DNA, double-stranded DNA, occurs in a helix, keeps it safe. During replication, we put it into a very unsafe position in order to develop a second double-stranded DNA set. Replication is going to be 100% of everything in that DNA needs to be copied. And DNA needs to be high fidelity. So we're going to talk about the difficulties of replication and specifically the replication fork. Learning all the enzymes that allows for the DNA to become unwound, copied, and protected while it does, and then also how DNA repair happens. Damage to the genetic code, errors, cannot be tolerated in DNA because any duplication of that error will propagate down a cell line. Even a prokaryote can't tolerate it, let alone a human. That's how we get a lot of genetic disorders. But from replication, more DNA is made. Right? And DNA is special in that it copies in the entirety of the DNA sequence. But a neuron does not make alanine aminotransferase, as the liver does. Skin on the toe doesn't produce insulin, and yet that genetic code is in its entirety in all places. So what we want to talk about is how do certain cells allow for certain genes to be transcribed, to be made into proteins, while others don't. How does that skin cell know it's not in the pancreas, and the pancreas does? Well, that's going to be based on the packaging of the DNA that's not necessary in the cell that it is. But that DNA that is necessary is going to be available to be accessed and turned into RNA. And if what we're making is messenger RNA, the process is called transcription. A copy is made of the DNA code to RNA to be sent elsewhere to be managed. Now, transcription is actually probably the most difficult thing we're going to cover. We're going to cover prokaryotic transcription as well as regulation. Then we're going to cover eukaryotic transcription and regulation. Transcription, turning DNA into RNA, is also going to bring about the construction of ribosomes, ribosomal rRNA, tRNA, mRNA, and in eukaryotes also some special types of RNA like snRNA that allows processing of that message. The idea is that we're going to send out specific copies, specific pieces of the DNA code into the cytoplasm to be translated into proteins. And pancreatic cells have pancreatic DNA exposed for RNA, whereas the squamous cell epithelium of your toes have different genes, and they do not include the production of insulin. Once we have our RNA, we then need to take what we've got in that information and turn it into an amino acid sequence. This process is by which we translate the genetic code. This is going to be mediated by codons, 3 nucleotides long. Transcription is going to be mediated by RNA, by the nucleotides A, G, C, U. Whereas DNA is going to consist of only 4 nucleotides, A, G, C, T, differing from RNA only by the T and the U. The codons will be 3 nucleotides at a time, translating the transcript, making whatever protein the DNA said to by a 2-step process. When we discuss translation, we'll actually discuss the steps of translation. What happens when you get a mutation, keeping it about point mutations, not about the complex stuff we'll get into medical genetics, and also what happens to those proteins to make them proteins, the post-translational modification. Because a simple linear sequence of amino acids does not get you a functional protein. Instead, those amino acids have to become full-fledged proteins by folding together and by combining their subunits. And then in order to fully understand amino acids, we are going to have to get into a little bit of enzyme kinetics, learn about the amino acids themselves, and we'll finish off with how to take the test in discovering the Lineweaver-Burke curves. Don't worry about that one. It's a bonus at the end. Really what I want to talk about here is why is eukaryotic DNA so special? Well, a eukaryotic organism, like a human, has a bunch of cells, and these cells have nuclei. And they have other specialized organelles that do things, like package proteins, send them outside the cell. Eukaryotic DNA is already, at its very baseline, protected. If you compare that instead to prokaryotic DNA, who have no nucleus, who just have their DNA, still double-stranded DNA, floating around in the cytoplasm, and no organelles to speak of. What you realize is that these prokaryotic organisms can only be bacteria. They can only be single-cellular organisms. And so all of their genetic material is found in this circular DNA. And because there's no impetus to have more DNA, there's no impetus in order to store this any better. What this allows for is replication to occur along the whole ring, but more importantly, what it allows for is transcription to occur from template strands in the same compartment as translation, which means that as a transcribing mRNA strand is made, ribosomes are working to make those amino acids. Eukaryotes are different. Pancreas, liver, toe, heart. How does the cell know to be which cell it is? Because there's a nucleus that can pack a lot of information in there, and because we have organelles, we're able to compartmentalize how we use our DNA. Single-stranded DNA does not last very long. In fact, exonucleases and endonucleases will eat up the RNA unless it's protected. It's tagged with a special set of proteins on either end, and eventually it will be degraded. The prokaryotes had a double strand to protect those base pairs. The first step of what eukaryotes did was having double-stranded, twisted helixes. This allowed in 3 dimensions for the nucleotides within to be protected. And this is the state in which most of our proteins can interact, unzip it, access what we need. Either get a protein like a RNA polymerase in there to make a transcript, or to start a DNA polymerase in order for replication. But if it kept just spinning around on itself, it would eventually get really tight and snap open. Right? Supercoiling isn't very sturdy. So what our bodies learn to do is take a protein called histone. And a histone consists of a couple of parts. 4 parts times 2 means that it's an octamer. And that octamer allows the DNA to get around and bind very tightly to the histone. This is made possible by arginine and lysine, positive charges that grab the DNA really close to it. Now, a DNA that's being bound by a different protein certainly is going to have trouble to be accessed by a protein that wants it. But if you look at this, it's not very efficient. If the entire genetic code for every cell has to fit in every cell, these histones are just going to get in the way. So what ends up happening is that if you put a bunch of these histones in a row, the double-stranded DNA wraps around these histones, but loosely. This loose connection is called a nucleosome. And a nucleosome is 10 nanometers in size, small, but able to be accessed. Each of these segments of this DNA between the histones, remember this is actually double-stranded DNA, right, is able to be accessed. But open DNA, stuff that's not packaged, it takes up a lot of space, is usable DNA. That is, it can be transcribed, it can be replicated. But it is also unstable. That is, exo or endonucleases can attack this. It's good and bad. Well, we can degrade it, get rid of it, the cell is dead, or we can fix it if there's problems with DNA synthesis, or we can use it for RNA. Again, though, this doesn't seem like it's saving a whole lot of space. So if you add histone 1, this signal, what it does is causes these histones to get really close together. And now not only are they forming a line, but they're forming a 3D structure wrapping around each other. And what happens is the DNA is wound so tightly around the histones and themselves, it becomes this mass of inaccessible DNA. This is a 30 nanometer band. The thing is, everything to the left is what makes a cell a cell. It allows for variation within the cell. Because you can imagine if you simply remove the H1, it goes back to this 10 nanometer easily accessible DNA. And so euchromatin is the chromatin that consists of DNA that allows us to transcribe and turn into proteins. When you're a pancreatic cell, you have nucleosomes, 10 and 30 nanometer fibers of your pancreatic DNA, pancreatic genes. The complexities that get this even more tightly wound up are probably not worth going into, other than to say that it becomes heterochromatin. Heterochromatin is all of that DNA that makes up every cell, but isn't needed or will ever be accessed by this cell. If you create these scaffolding proteins and you take these 30 nanometer fibers, you can have little specks along the way. But you start tacking on these specks and all of a sudden these great structures of these 30 nanometer fibers, conglomerates form. And you put those together and wrap them together, and what you get are these giant egg-shaped things where you can no longer see the scaffolding proteins because they're so deep inside. And there's basically no way to get this DNA in at all unless there's replication happening where it finally opens up. The idea is that there is going to be DNA that the cell will never need access to. It's thick, it's obviously spotted on an electron micrograph. It's the dark black bands. But where it's nice and little thin bands are going to be the euchromatin which could be accessed to make proteins by making RNA. Because eukaryotes have organelles in the nucleus, they separate each of these things. DNA and replication is kept in the nucleus. RNA is made in the nucleus, processed there, but then exported to the cytoplasm where ribosomes encounter it, and then it may end up in the Golgi through the endoplasmic reticulum, or it may just simply end up in the cytoplasm. It certainly can't happen at the same time. But prokaryotes don't have these restrictions because they don't do very much. And there's never a state in which a prokaryote turns into another cell. In the eukaryote, every cell can turn into another cell. Once it's differentiated, it's locked in its cell type, okay. But it all came from a pluripotent fertilized egg that could have become any cell, and the genes tell it along the way. When genes aren't needed anymore by a particular cell type, they're still in the cell, they're just tacked away. So this was an overview. Here's the course, start to finish, left to right. Here's the cool features of eukaryotic DNA. We're going to get into a lot of details and things you need to learn. This was just more of an overview and introduction. That is an introduction to the biochem DNA to protein.